So thank you again, Jack, and see you soon.
Thank you very much.
Thank you, bye. So the next talk will be given by Rosen Mingim, and she will talk about data visualization. Rosen, are you around?
Yes, can you hear me?
I can hear you, and I can see your screen, so it's perfect.
Great. Thank you very much. Thank you very much for the attendance and the conference to have accepted our abstract. I thought, I've been interested for quite a while in this application of Soundscape ecology, or whatever you want to call it. And I thought it might be interesting to have a little bit of the perspective from the point of view of the data scientist or the computer scientist in a conference like that. So the work that I'm presenting today, there's more things that we did and more people involved, but these are the main people involved in the work that I'm presenting today. Many of them computer scientists, some of them in the application area, like ecologists and zoologists. And the institutions involved are basically 3 universities in São Paulo state in Brazil, and where I worked in one of them, University of São Paulo, until 2 years ago, and now I'm in Cork, Ireland. So basically, our intention was to try and understand the problems, some of the problems, and try to look for solutions in terms of handling these very complex type of data. One of the complexities coming from Soundscape ecology is the fact that one type of data, meaning the sound that is recorded in the environment, plus of course other things that we can collect, such as weather and all sorts of environmental information, that one type of data is meant to solve a whole range of different problems. So this is something that in data science, machine learning, or like people like to say AI, is a common thing that happens, because quite a few data sets are actually representing different phenomena in the same sets of attributes. But anyway, so we started looking into segregation tasks and the role of feature selection and active learning in the process of helping people in the application area to handle the data, and then the additional questions that they have about the environments they are studying. So in terms of segregation tasks, we've handled direct feature extraction, such as we've been seeing in many of the works that were presented in this Congress. And also we've looked into neural models to solve some of the segregation problems. So I'm talking about a little bit of those things and the next steps in our work with our partners. Okay, so we can see that data science, machine learning tasks have been permeating the work of Soundscape ecology, ecoacoustics, looking at the sample of the work that we've attended to in this Congress, we can see that's going on quite a bit. So we wanted to know what we could do to help, right? So demonstrate the support of such techniques in environmental studies. So our first approach was to build supportive tools to define discriminative features, meaning trying to study what kinds of things we can obtain from the data that can help discriminate certain events or certain phenomena happening in environments in these recordings and registered by these recordings. So, and also develop methods for automatic and interactive tasks. So right now, we still don't know how to model, how to build good models for certain tasks in this environment. So we want to first study how we can extract information from that. So that's the point of view of the computer scientists. So we started by trying to discriminate different things from audio recordings, starting with different landscapes, then different groups of events, and then different species. Okay? So in all of those, we used various types of visualization, but the most common one that reappears, and we can see many of the work that we saw in this Congress, is what we tend to call multidimensional projection, but the people also call embeddings, dimension reduction, okay? Which is you have a data set, you actually pre-process that data set, you extract a number of features from that data set, and then you do some kind of mapping of that data into 2 dimensions or 3 dimensions, it could be the case, to try and find how these elements group in terms of similarity. Okay, and that is an indication that the original extracted features can help discriminate certain things inside your data set. So we have seen people using quite a few of these projections in the work that we've seen, but there are plenty of different ways of doing that, such as PCA, LSP, TSNE, 4-space layouts, and UMAP, some of which we have seen around in the last 2 days or 3 days, including today. So in the first, so what I've been calling these different types of things that we are looking for as scales, right? So in the top scale, we have the distinction of landscapes. So this was a study that we did on that. We collected, we used data collected in different environments, and we want to try and find what types of features could help discriminate those environments automatically in the future. But at first, we would have to do some tasking processing and case studies. So this is the method. We from the sound signal, we extract acoustic indices as well as other acoustic measurements directly from the sound or from the actual image of the spectrogram, right? So from the spectrogram image, we do standard descriptions and also unsupervised learned descriptors. I'm going to give some detail, but more detail we can talk later. So on that, we do visualizations and also numeric evaluation of the power of discrimination, of segregation of these different quantities, these different features or attributes that we extract from the sound to acquire knowledge. So a projection then gets all the different extracted elements for the different audio recordings. And at some point, we do have labels for at least part of the data set, a good part of the data set, so we can test our approaches. And then a projection actually maps that into 2D. It's like you are doing a PCA, only there are techniques that are more bound to find segregation in terms of different attributes, okay? So in this first experiment, we had files from 2 different forest environments in Costa Rica. We called Costa Rica 1 and 2. They were 10 minutes and 1 minute in length, and we processed as they were, okay? So, and we also had 2 underwater environments in 2 different places in Brazil. So we calculated various acoustic indices, various image descriptors, and various measurements directly from the audio, okay? In total, it was, Fabio is there, but I think it was 130 around that attributes in the end. And then the combination of that with this in particular is TSNE projection can show that, that we can separate those environments, okay? Also, there is a mixture of two environments here because there were two different collections in alleles, but they are actually not distinct in terms of any of the aspects from the biological environmental point of view. So it was expected that they would mix anyway. Also, we examined some of these groups that form, and we did find some consistency in the types of sounds that are grouped together. So in the second scale, we tried, okay, so what do we do if we want to distinguish different groups of events? In this case, we worked with frogs, birds, and insects. So what if I have a bunch of files and I want to tell where they are? Which ones are frogs, which ones are birds, and which ones are insects, okay? So we did some work on that. And for that, we felt that the same solution for environments, for general landscapes, would not work in that kind of scale. For that, what we did, we extracted features, all of those plus a few more, and we developed a process for selecting relevant features using standard relevant feature algorithms, okay, feature relevance algorithms. In this case, extra tree classifiers. And then we validated the selection using different classification methods to reach a certain feature selection strategy for separating between groups of events, okay? So the experiment was in an area in São Paulo State called Cantareira Mantiqueira, and with different cover of vegetation, and we had frogs, insects, and birds, and again, acoustic indices, image descriptors, and measurements from the spectrogram. And in this case, we did find some results that managed to distinguish between those groups of events with higher accuracy. Now, when we get to scale 3, I'm not going to give details because it's a lot more complex than that, than the time allowed, and I know I'm ending my time here. But when we try to describe or to discriminate species inside the same environments, we had to resort to neural network models, which we have been using quite successfully, I would say. So basically, those that also use quantification as a way of feeding back into the process to improve data collection. So the neural network models actually extract features from these environments, and which would be step 1 here in creating the visualizations, right? And then this is the difference. The lower line here is when we calculate the features, and we use those features to try and discriminate the data. And on top here is the actual output from the neural network. So we can actually see, even without the numbers we use to find the level of segregation, that the neural network models are much more successful than the actual initial features that are directly extracted from the data. And then we are building something like this. This is a tool for feature selection. So here is an example of birds and insects. The name is there somewhere. Is 2 of those 3 groups of events. And here is the features that are extracted. So the user comes here. This visualization means something. We can talk about that later. but we can use automatic feature selectors, and then the user can go there and select by themselves. I want ACI, I want biodiversity index, I want whatever to add or to remove from your set of features. So for instance, from 143 features here, we have 11, and there is already a very nice separation starting from these two groups of elements. And this is the process. I'm also not going to go into detail. The other thing that we are working on is active learning for labeling. So how do we capitalize in these types of results to help users label and to suggest labeling for the experts, so it is sped up. The process both of labeling for processing and for understanding of the environment. Okay, so that's more or less it. I do have some more, but my time is over. And I'm actually editing a special issue on visual data mining. So if anybody is actually working on data mining, visualization, machine learning processes, applications are welcome. So you might consider submitting to it. The journal is algorithm. Okay. I thank also some other colleagues for data that was used in this work, and also very interesting discussions in terms of the results. Thank you so much. Sorry, I overstepped a little bit.
No, it's fine. Thank you very much, Rosie. So very interesting, plenty of information, plenty of results. Brilliant. Are there any questions to Rosie? I would have one, but maybe I missed it. I'm sorry if I did. Which kind of neural networks did you use?
No, you didn't miss. It's just that that there was no time to go. Okay. We did conventional CNNs. Okay. We trained ourselves. We did CNN with dropout. We did CNN with quantification, and we used predefined, pre-trained networks such as ResNet 50, which is pre-trained in images. And we feed the neural networks with the actual spectrograms so far. Okay. And then the best results so far were our own trained networks with quantification. But of course, our domain was still restricted in species of frogs and birds all together. Okay.
Very nice. Yes, thank you. And which language do you use?
Well, the visualization part is usually JavaScript. Underneath it's either R or Python.
Oh yes, okay. Python, I guess, for the CNN. No.
Yes, one of them was Python, the other was R.
Okay. Any other question? Let me check. Okay, Klaus Rieder is asking, is doing the further remarks that differentiating some frogs and insects, in particular crickets, can be really hard. How many samples did you test and validate?
Yeah, good question. Okay, let me get here the actual, I'll do it in just 1 second. This is the frog thing. No, actually, just a second, I'll get there. So this is actually the system running on the output. Oh, here. Let's see if I can help this somehow. No, it's not going well with my, but anyway, this is the actual system running with the output of the network on the 12 species. Okay, so basically the output of the network, now we don't know what they are. So when we extract features, we see here age, bio, ACI, and so on. But here, we don't know anymore because it's the output of the network. So I select here and I see it here. So the species we can actually see the species here when we are looking at it. What was the specific question?
There was the size of your training and validation.
Oh yeah, the size. Okay, so we had 6,780 labeled points, and let me get here the actual numbers. I'll see in a minute. I had to select in the end. If Fabio is there, he can answer.
Okay, but maybe you can answer in the chat just afterward if you do not.
Yes, I can do that.
Okay.
I'll let you know. It's in the order of 600 to 800 per species.
Well, okay, we see this more or less the scale.
It's a good scale, yeah.
Okay, so thank you very much, Rosanne. We need to go on to the next speaker. Thank you very much.